My grad student, Miss Linda Major. Linda was an undergraduate here some years back, and I remember taking the developmental biology course with me. And during that, she was especially fascinated by some of the mechanisms that we just briefly touched on. Things like apoptosis, things like especially epigenetic changes that happen in the histones and their effects on gene regulation, transcription, what have you. And when she became a graduate student, she wanted to see if we could do something like that. So she'd be piling papers on and looking, oh, we don't have these kind of facilities, you know, most of them are big research universities. But she discovered a paper in PLOS One by Finnish group. Was that Finnish? Headed by somebody named Cascone. And she said, hey, can we do something like that? Well, not at that level of sophistication, but we could strip it down and it would be amenable to a master's project. And she quickly put a proposal together and then plunged into the literature and finally plunged into the lab and did I don't know how many, finding all the fine points of how you can have problems with plant cell fractionation, which explains why almost everybody is an animal cell biologist, trust me. So many, many late nights, 2 a.m., 3 a.m. nights in the lab to finally get her organelle fractionation procedures to work for this system she was studying. And then we started getting some preliminary but very intriguing results. And this, what she has come up with, I think is really going to direct the research, at least in my lab, and keep many students busy for a long time to come. It's opened up a ton of cool avenues that we can investigate at a preliminary level, and maybe we can get some money in or something like that at a more sophisticated level. Anyway, I would like to leave it to Miss Linda. She's got an interesting story to tell. So, Linda, rock and roll.
Okay, my research is based on the idea that a cell's genome is continuously exposed to agents that damage its DNA. Damaged DNA activates the cell's damage response to either repair the DNA or induce apoptosis, which results in the demise of the cell. During the DNA damage response that induces apoptosis, chromatin compaction around the damaged site is relaxed through epigenetic core histone modifications and by displacement of the linker histone. At the beginning of 2012, Cascone and her team published a study that showed in animal cells displaced histones enter the cytoplasm and bind to the cell's mitochondrion that leads to the permeabilization of their inner and outer membranes. This permeabilization causes cytochrome C to be released from the mitochondrion's intermembrane space. The following few slides are background information that's pertinent to this, to my research. Here, you'll see a cartoon of mitochondrion. This outer membrane is the membrane that I believe the mitochondria bind to. The inner membrane is where the electron transport chain resides, and the space in between the outer and inner membrane is the intermembrane space where cytochrome C normally resides. Cytochrome C is a very interesting small protein. It's approximately 15 kilodaltons in molecular weight. It's hydrophilic and with a central heme group that allows it to transport electrons from the third complex in the electron transport chain to the fourth complex in the electron transport chain. For whatever reason, for some reason, for the reason that cytochrome C gets out of this intermembrane space and it is found in the cytoplasm of the cell, this cytochrome C becomes the commitment molecule in the apoptotic pathway. Apoptosis is the cell's programmed death mechanism. There are two pathways in the apoptotic process. The one pathway is the extrinsic and the other pathway is the intrinsic. The extrinsic pathway is initiated by external factors such as toxins. This pathway uses the death receptors in the cell membrane that activates a mitochondrial channel in the outer membrane of the mitochondria that becomes activated, and it transports cytochrome C from the intermembrane space to the cell's cytoplasm. And the intrinsic pathway is initiated by internal factors such as radiation, embryonic development, cellular development, and cellular aging. This pathway is initiated by binding of displaced histones to the mitochondrion's outer membrane that causes cytochrome C to leak into the cell's cytoplasm. My research deals explicitly with the intrinsic pathway and not the extrinsic pathway. The goal of my research is to work with plant material to determine if the results of Cascone's animal study are seen in plants. And the questions that this research will answer are: first, do mitochondria bind to histones bind to mitochondria in plants? And if the answer to that question is yes, try to answer the question, do the bound histones affect the release of cytochrome C from the mitochondria? And then based on the answer to the first question, do histones bind to mitochondria, I attempt to answer, do histones bind to chloroplasts? The methods I use to conduct this research include the isolation and purification of mitochondria in histones, confirmation that mitochondria are viable, and confirmation that histones were isolated. I used cauliflower. I ground it up, filtered it. I used differential centrifugation to isolate mitochondria and nuclei. I further isolated mitochondria using Perkal density gradients and washings. And then I used DCIP to test the viability of the mitochondrial organelles. DCIP is an electron acceptor that intercepts the electrons as they're being passed down the electron transport chain. If the electron transport chain is working, if it's intact, then the DCIP will, as I said, intercept this electron and change the blue color of the DCIP to clear. And as you see here, that did happen, so these mitochondria are considered viable. Then I further isolated the histones using a high salt extraction protocol that strips the histones from the chromatin. And I confirmed this isolation of the H3 using Western blot against H3 antibodies, which you see here that there is that evidence. So I considered, it's considered that the histones have been isolated. The procedures I use to answer the first and second question, do histones bind to mitochondria, and if they do they affect the release of cytochrome C? It's very simple. I had my histone fraction, I added to my mitochondria fraction, I incubated them for 10 minutes, then I centrifuged the mixture to separate the pellet containing the mitochondria with the bound histones from the supernatant containing the histones that did not bind to the mitochondria. I used the pellet and the supernatant to provide evidence to show if histones bind to mitochondria by running a Western blot against H3 histone antibodies, and also to provide evidence to show if cytochrome C has leaked out of the mitochondria bound with histones by running a Western blot using cytochrome C antibodies. And so to answer the first question, do histones bind to mitochondria, what I have here is the resulting Western blot, and this is the molecular weight protein standard. This is the 15 kilodalton band, where both the histones and the cytochrome C will reside. This is against the H3 histone antibodies, and you see here in the histone control that I did isolate histones. This is one mitochondrial sample, and this is a second mitochondrial sample. You'll notice that there are no histones bound to these two samples, showing that they are clean samples. These two lanes, I incubated the histone control with the first sample, the histone control with the second sample, and this is the result, the pellet and the supernatant. And you can see here that there is evidence showing that histones did bind to the mitochondria, and again here the histones did bind to the mitochondria. This residual here shows that I did put in an excess of histones, so whatever did bind, whatever could bind to the mitochondria did, and there was obviously some mitochondria that had no place to bind to, therefore they remained in the supernatant. And you can see here that this mitochondrial sample was had a higher concentration of the mitochondria than this sample. So to answer this question, do histones bind to mitochondria? Yes, they do. Now for the second question, do bound histones cause the release of cytochrome C? First I'll explain what I have here, and then we'll go on to the results. This is the Western, these are two Western blots, two pictures of two Western blots. This top one is the Western blot against the H3 histone antibodies, and the bottom one is the Western blot against the cytochrome C antibodies. This is the molecular weight protein standard. Again, as I said, the 15 kilodalton range, which is this blue band, are the proteins of interest. These proteins here are just simply proteins that cross-reacted with the cytochrome C. They're of no interest to this research. Okay, as I said, these are just the two bands of interest. Again, it's the same concept. I have the histone control. I have the histone control and the mitochondria control, and then the last two lanes are the mitochondria incubated with the histones. And you'll notice in lanes 2 and 3 that there is evidence of histones in both the pellet and the supernatant, which with much more evidence of histones in the supernatant. You're going to see some residual histones in the pellet for a couple of reasons. One, they probably bind to the plastic of the tube, and second, it is just no conceivable way to suck up every last bit of liquid out of those tubes. Now you'll notice that there is no evidence of cytochrome C in either the pellet or the supernatant, and this means that this histone fraction, I consider this histone fraction analytically free of mitochondria and any mitochondria-specific protein, cytochrome C. What you have next is lanes 4 and 5, which is the mitochondrial control. And you'll notice that there is evidence of histones in the pellet and not the supernatant, which shows that some of the nuclei will rupture during the mitochondrial isolation procedure, and whatever histones were released actually did bind to the mitochondria. And you'll notice that there is no cytochrome C in, I'm sorry, there's evidence of cytochrome C in the pellet, but there is no cytochrome C evidence in the supernatant, which means that there is still plenty of intact mitochondria in the sample. And what you have lastly is the lanes 6 and 7, which are the mitochondria with histone incubated samples. You'll notice that there are histones in the pellet and supernatant, and much more histones in the incubated sample here than here in the control. This shows that, again, that there were an excess of histones added to the mitochondria sample, that whatever histones could bind to the mitochondria did, and whatever was left over remained in the supernatant. But now you'll notice that there is cytochrome C evident here in the supernatant that was not evident here in the mitochondria. And the faintness of this band is simply due to the dilution of the supernatant. Cascone also had that problem, and they attempted to concentrate the supernatant, which I did not do. So in answer to this question, do bound histones cause the release of cytochrome C? Yes, they do. So now back to the third question, and you need to ask, why is this of any interest? First, both mitochondria and chloroplasts are organelles. Second, both of these organelles have a double membrane. And third, both of these organelles are the result of a symbiotic relationship. So it just begs the question, do histones also bind to chloroplasts? The methods I use are similar to the methods I used in the first and second questions. I isolate chloroplast using spinach and Perkal density gradient. I confirm that the chloroplasts are viable using DCIP as the electron acceptor. And I confirm whether or not histones bind to chloroplast using Western blot against H3 histone antibodies. Again, this is just a quick pass-through pictorially of what I did. I used spinach, I ground it, filtered it, differential centrifugation to isolate. I further isolated using a Perkal density gradient and subsequent washings. And then I confirmed that the chloroplasts were viable by using the DCIP reaction test. And you can see the original chloroplast sample after DCIP is added, and then the resulting color change, which proves that, or confirms that the chloroplasts are viable. This is the SDS page gel of the experiments that I ran. You'll notice that the first and the last lane are two different mitochondrial samples incubated with the histones. The middle lane is the molecular weight protein standard, and these are three different chloroplast samples. This is a fourth chloroplast sample, just having a little different amounts in the gel. And here you have the Western blot results of this against the H3 antibody. You can see in the first and the last lane, which are the mitochondrial lanes. You have to excuse this. I cut my lane a little short. But you can see that there are histone, bound histones in these two mitochondrial samples. But you'll notice here that none of these four different samples, chloroplast samples, have any evidence of bound histones. So the answer to this question, do histones bind to chloroplast? I would say the answer is no, according to the results of this test. So the overall conclusions are, to questions 1 and 2, do histones bind to mitochondria, and if they do they cause the release of cytochrome C? evidence shows that histones do bind to the mitochondria, and that by bound histones cause cytochrome C to leak out of the mitochondria. This suggests that histones do affect the release of cytochrome C in plants, and that this histone and mitochondria interaction are highly conserved amongst plants and animals. The conclusion from the third question, do histones bind to chloroplasts? Evidence shows that histones do not bind to chloroplasts. And this suggests that histones do not have an interaction with the chloroplast, and that the histone interaction is not similar amongst these organelles. Future studies will include testing fungi, protists, and invertebrates to determine if histones similarly bind to mitochondria in these taxon, and to also investigate the extent of conservation by cross-incubating histones from one taxon with organelles from another. And with that, I thank you very much. I thank JSU for giving me the space and the money to do my research. I thank Drs. Hamasu and Klein for being my advisors, and well, I just thank Dr. Sauter for being Dr. Sauter to me.
I just did this.
And with that, are there any questions? Yes.
So you're saying histones, like tested H3.
Yes.
So what about H1 for the chloroplast?
Okay.
Did you look to see if there was any eruption with the chloroplast?
There was no, because once you.
Because you looked for cytochrome C.
Mhm.
So did you address that issue before?
Okay, a couple of things. In Cascone's study, different histones did different things. In their study, they were able to isolate H1 from H2, H3, and H4. I was not able to do that. So H1 had a higher effect on one of the membranes. H3 had a higher effect on the other membrane. Histone protein antibodies are difficult to come by in plants. One thing, I had to take what was available. Second, they're very expensive. So I could not purchase more than one antibody. So with that, it took also time. There were definite time constraints. All the procedures available for the isolation of histones, nuclei for that matter, for chloroplasts, for mitochondria, although they were documented in studies all over the place, did not work with, they didn't work. I had to change, alter, I had to figure out ways to isolate these various things, which is how I went from the green plant material to the plant material with no chloroplasts in it. So time constraints only allowed me to do so much. When it got down to the chloroplast, that was the last thing. Interestingly, it should have been the first thing, but I could not also isolate the histones from the green plant material. I could not isolate mitochondria from the green plant material. All I could do was isolate the chloroplast from the green plant material. So there are many questions. Is it because histones from the cauliflower don't work with histones from the spinach chloroplast? Is it because the H3 doesn't work? Maybe it's the H1, maybe it's H2, maybe it's a different mechanism. All those questions I thought of, but I ran out of time, I ran out of money, and I want to have things to do next summer. More questions?
You may have answered it in one you just did. When you looking at your histones, showing the histones binding the mitochondria, your pure histone fraction, at least as I recall from your slide, had 2 clear bands in it. When you said my histones and mitochondria and saw my histones in that band, it looked like there were 3 histone bands. And I just didn't, I did.
Tell me when to stop.
Let's see, it's,
Further back.
yeah. Oh. Yeah, there we go. So you got your histone here, I see 2 clear bands there, I see 3 clear bands over here.
Are you using your own little
I
pointer
didn't use
device?
my own pointer for that. My apologies.
Dueling
My
pointers here.
apologies for that.
Okay,
Yes, I-
are you talking about this? The 3 bands
Yeah, I'm talking
here?
about 3 bands right there.
Okay, what,
Sorry.
I'm saying I'm not moving my pointer. What we have here is something that is of interest to me. I hope it's of interest to you, and this is epigenetics. It has to do with epigenetics. Every histone, all histones have a very basic short tail that extends out from the core of the histone. These tails get modified through addition of methyl groups or acetyl groups, phosphoryl groups, and various other things. What you're looking at here is the H3 histones that have been epigenetically modified. Therefore, they're going to have a different molecular weight. Thank you for letting me just buzz that right in there.
What's the detection limit on your Western hybridization? Is it as sensitive as something like chemiluminescent, or is it not as sensitive? Other Western hybridization techniques, is it the sensitivity low or high, or what is it relative to like chemiluminescent?
I don't know the answer to that question.
I think in general, luminescence can often be more sensitive. The big problem with that is the signal lasts for about a minute. And then you have to I hate ECL. I've used ECL in the past and I can't stand it despite the sensitivity. So we're using alkaline phosphatase, which has a stable signal. If you use horseradish peroxidase, the signal disappears in a few days. You got to take those pictures immediately. I don't like HRP. Dr. Homsey uses alkaline phosphatase routinely too. So it's a little less sensitive to chemoluminescence and a lot less sensitive than radioactive iodine. We can't do that here anyway. I didn't want that.
You
More
may have
questions?
answered the question that this was in plants. Is there a similar mechanism in animals where the histones bind to the mitochondria?
That was the beginning of this whole thing. Cascone and her team, that's what they discovered, that histones bind to the mitochondria and affect the release of cytochrome C. So that's how
In animal cells?
they used rat. I think they used rat.
Liver,
I think they used the rat liver.
Yeah.
Okay.
Yeah.
At least it's true in mammals. They never tested.
Well, it just got me thinking. I'm wondering if there's a difference in things like immortalized cell lines, like cancerous cells. If there's a difference between the histone binding to stimulate apoptosis.
That's another master's project.
That's another.
Any more questions? I'm good with questions.
So just for my own
Yeah.
general knowledge, these are binding to the outer membrane, but now you have cytochrome C coming out. Are there any of those histone components that they know that bind to the inner membrane?
Absolutely, yes.
Is it
I
H1?
I want to say H1, but I'm not positive. But there is definitely a difference. They all bind to both the membranes. Just like one has a higher affinity for this membrane. They found it more in this membrane than this membrane. They found another one more in the inner membrane than the outer membrane. And I also can't tell, did it only bind to the outer membrane or the outer and the inner membrane? There's no way for me to tell that. I could just tell that it bound to the outer membrane because the cytochrome C was inside the intact mitochondria, and after I incubated it with the histone, it was no longer inside the intermembrane space, and that's all I could tell. Going. Applause. Thank you very much.
Thanks for coming, people. Thanks for coming, and any of you, darn terrorists.
Oh yeah, I forgot to turn it off. I didn't tell you that.
Well, folks, anybody here looking for a future master's project?
We have, I know, okay, yeah.